polymorphism. Already I gave you definition. If anybody is having any doubt regarding this, you can ask here. DNA polymorphism is the polymorphism when it is seen in more than one allele at a locus in a population with a frequency greater than 0.01, then it is said to be DNA polymorphism. Then when the frequency is greater than 0.01 only, we say it as a DNA polymorphism. Below 0.01, we don't consider it. Then in simple terms, how can we define? So any mutation which is occurring in germinal cells, germ cells, which is inheritable and which is inherited with a high frequency. This is what is very important. Then we say that as DNA polymorphism. And these kind of the probability of this variation is mostly seen in non-coding DNA sequences. As mutations in these sequences cannot be having any effect because they are non-coding now. Mutations occur, but not showing any effect, immediate impact. Impact immediately is not there. And they will not affect the reproductive ability of the organism also. But these mutations keep on accumulating, generations to generations, and leads to evolution. Right, we have discussed in Human Genome Project single nucleotide polymorphism. That is one type of polymorphism. Now I'll come to the point. That is a type of polymorphism. So polymorphism or variability is of various types. Starting from single nucleotide polymorphism. This is the base single nucleotide change. 1.4 million locations we show single nucleotide polymorphism and the frequency is greater than 0.01. That's why we said it as polymorphism. 1.4 million locations we show SNPs. We have single nucleotide change. And all these polymorphisms somehow are leading to evolution, are leading to speciation, formation of new species. Now, at this level, this is the idea of polymorphism. And basically, if I once again run this particular concept with the help of the questions, you please answer. Now, what is polymorphism? Anybody try to give a definition of that? So, and I will be very happy. Aishwarya, can you give me what is just polymorphism? In whatever way you understand, you let me know. Aishwarya? Ajitesh?
Variation in the genetic levels produced due to mutations.
Then Semoli, can you tell me what is DNA polymorphism? Sadhana, will you try? Whatever you understand, only that thing I want.
Variation in the allelic sequence of DNA.
Variation in the allelic sequence of the DNA. In only one allelic sequence or more than one?
Less than one, sir.
More than one or less than one? Check out here.
Sir, greater than, sir. Greater than one.
More than one. More than one allele. Allele, you are talking about allele? More than one. And its frequency in a population is how much?
Greater than 0.01.
Then that is called as DNA polymorphism. Polymorphism is variation. And variation in more than one allele at a particular locus. And that particular variation in a population frequency greater than 0.01, then we describe technically it as DNA polymorphism. Geetika, can you tell me DNA polymorphism definition in your own words without confusion?
Sir, if an inheritable mutation is observed in a population where more than one allele at a locus is being no sir, 1 minute, sir. Sir, no sir, I do not know how to verbal it, sir.
Okay. Right. So you want to choose this definition or this definition? There are two definitions for it. Which definition you want to choose?
First definition, sir.
First, what is that? Can you please help me?
Sir, if polymorphism at more than one allele at a locus in a population with a frequency greater than 0.01 is called DNA polymorphism, sir.
Then what is this definition? What it is saying?
Same thing only, sir.
Same thing, but what is it? What I have added here?
Inheritable mutation, sir.
Inheritable mutation. So inheritable mutation is seen in population at higher frequency. Now, what is the frequency that is given in the above statement? Right, very good, thank you. So this is what DNA polymorphism. Then these, as I told you, these polymorphisms are of many types. Right, they are of many types. Like, let us see what are those many types. In that, we have already seen single nucleotide change. So let me write some more important features of this polymorphism. I am not going out of your text. I am completely within the line and helping you to understand what is the concept, right? So no need to worry about this. I just 2-3 extra points.
Sir, in the first definition you have written polymorphism in more than one allele at a locus, no, sir?
Yes.
But in a locus, only one allele will be present, no, sir?
That particular a locus consists of one allele, right? Another locus consists of another allele. Like one allele at a locus means each and every locus only one allele will be there. I said you have to take from here as a different statement, Geetika. That's why I have given the syntaxes which you cannot able to understand in the text. I have taken the same syntaxes. If more than one allele at a locus means one allele at a locus means you have to consider this whole bracket. Means 2 alleles at 2 locus, 3 alleles at 3 locus, 4 alleles at 4 locals. Got the idea? 4 locus. 5 alleles at 5 locus. More than one allele at a locus, more than 2 alleles at 2 loci, more than 3 allele at 3 loci. You understand this? This whole statement is related to, as one unit. One allele is present at a locus. If it occurs more than one allele at a locus means 2 alleles. 2 alleles are present at one loci or 2 different loci? How many? 2 different loci. That means more than one allele. It may be 2 alleles at 2 loci. It may be 3 alleles at 3 loci. It may be 4 alleles at 4 loci. Right? It may be 5 alleles at 5 loci. But it should be more than what? One allele at a locus. He is just telling you a allele is present at a particular loci. Clear?
Yes.
That one allele means one variation. I say there is one variant. One variant means at a particular locus. At a particular locus, what is present? Allele, sequence of the gene. Then if the polymorphism, if I write like this now, if polymorphism is more than one allele at a locus, it may be 2 alleles, it may be 3, it may be 4, it may be 5. You can take more than one means maybe 2, 3, 4, 5. And each allele will be present at their specific locus. If you are taking 2 alleles, they are present at 2 loci. 3 alleles present at 3 loci. That is the meaning. You understand this?
Yes, sir. Sir, if we are removing the word at a locus, will the definition change?
Nothing will change. You want to simplify, you can simplify it more. If polymorphism is more than one allele in a population with a frequency greater than 0.01, also DNA polymorphism. But to indicate that each allele is present at a particular locus, he has given one allele at a locus. So I'm just helping you to make it clearly understand. You can also write if polymorphism is more than one allele in a population with a frequency greater than 0.01, then that is DNA polymorphism. And this can be simplified like this DNA polymorphism is nothing but the inheritable mutation which is seen in the population with higher frequency is nothing but DNA polymorphism. Ultimately, the two statements, different statements which are there, which are given for you, indicates that the variation in the allelic sequence, not at one place, more than one place, is inherited and seen in the population with a frequency greater than 0.01 is DNA polymorphism. Now you can combine this. So at the end of the class, you have to write about in your own terms what you understand about polymorphism. This is the homework. Right. You understand Geetika now? Shall I move on?
Yes, sir.
Okay, let's move on. So this is about polymorphism. As I said, polymorphism is of many types. Right? In that, single nucleotide change, this is one type. Polymorphism is of different types. And they usually range from single nucleotide change. They usually range from single nucleotide change. This is the basic fundamental feature, single nucleotide change. We already discussed about this. 1.4 million locations repeating again and again. And this polymorphism are useful for speciation, means formation of new species, and evolution. Right? So this is done. So any doubts regarding this, you can. So everything is fine. So those who have got the clarity of polymorphism and what we call it as repeated sequences, please raise your hands. Then we'll move to the next concept. Methods or technique of DNA fingerprinting. Very clear about the concept, only raise your hands. Otherwise, I will explain once again. Abhirami SK, Abhirami M, Achyut, Aishwarya P, Ajidesh. We'll discuss again if you do not understand. So you have to show the consent. Abhirami SK still not raised their hand. 18, only 18 students. What about the remaining? Are you there in the class? Abhirami M. Only 27. What about the remaining? Gautaman, you understand or you have any doubt? Gautam? Gautam, are you there?
Sir, understood, sir. Understood.
Jay Anirudh? Okay. Genovia? Jay Anirudh, am I audible? Right. Genovia? Right. Okay, we'll move on. Let's start the procedure. Okay. Technique. This is developed by Alec Jeffrey. Right. Now let us write down some of the some other scientists' name also. Otherwise, we will directly jump into the concept. Just a minute. The board is visible to you?
Yes, sir.
Right. We'll start the concept. The first step in the case of this DNA fingerprinting, as usual, isolation of the DNA. Step 2, breaking the DNA or digestion of the DNA by restriction endonucleases. Restriction endonucleases are the enzymes which cut the DNA at specific locations in between the DNA, not at the ends, right in between the DNA. They cut the DNA at specific locations, not random locations. They specifically identify a sequence and cut the DNA. Then separation of the DNA fragments by electrophoresis. Step 3.
Sir, previously, sir, about polymorphism, sir. Sir, a polymorphism can range from a small change in nucleotide, sir, to any changes, sir.
To bigger changes.
Yes, sir.
It depends upon the intensity of the mutation.
Yes, sir.
Intensity. How much intensity? If it is high intensity mutation, it leads to larger change. Right. And these changes are more seen in satellite DNA. It was observed that the satellite DNA has undergone a lot of changes over a period of time, not what we call it as not in a very short span of time, because you know the DNA is only shows slow rate of mutation. Here various knowledge you have to apply, because we have already discussed about the DNA. It shows slow rate of mutation. That is one of the stable character of the DNA. That you don't think that sir, polymorphism is seen, so mutations are occurring in the DNA, so they are inheritable. So we should see a lot of changes very fastly. No. The changes are very slow because the mutation rate is also slow. Rate is slow. Frequency is greater than 0.01 in a population. So it is DNA polymorphism. When that particular mutation frequency is greater than 0.01 in the population, then we say it as DNA polymorphism. We say variation in the DNA. Then there will be so many variations, but if they are not distinct, they are distinct when they cross this frequency 0.01. Then we say a clear-cut variation. So these DNA polymorphisms, which are with a frequency greater than 0.01, that is called as high frequency, are seen in the humans. Then they have accumulated from lots of generations. They have passed on from so many generations. Then they became they are shown as DNA polymorphism. Right? Is that clear?
Then most of the most of the evolution is due to changes in the satellite DNA.
Yes, let's say you can interpret most of the changes, because of the changes in the DNA. See, that's the reason. Still, we don't know. That's a good question. Then I will take some time. See, because of the changes in that satellite DNA, maybe it leads to evolution and speciation. The researchers are saying the same thing. But at the same time, parallelly, they were saying that they are non-coding regions of the DNA. You understand, Ajitesh? At the same time, they were saying satellite DNA are non-coding regions of the DNA. Okay, then parallelly, they were saying these mutations accumulated and helps in evolution and speciation. This is the general terms they have given. As you go to the higher classes, you will understand the impact. And we only, I think we know 5% of the genes that we have discovered. I think 5% or I think 20% we have studied in Human Genome Project. Only very less amount of genes. The remaining genes, we don't know the functions. Still, the functions are unknown. Still, exactly, we don't know what is the importance of these repetitive sequences. See, this is during the research, one has found out that yes, there is a difference in the DNA of one individual to the other individual in the repetition sequences. Then immediately they used these repeated sequences in the satellite DNA as to identify the difference between two individuals. It has started with that. Then if you talk about the polymorphism, that's another subject, because this subject will be dependent only on the DNA fingerprints and repeated sequences. Then he has also given you an idea of polymorphism, that polymorphism is of many types. Variable number tandem repeats is a type of polymorphism. Single nucleotide change is a type of polymorphism. Change in allelic sequence at 5 or 6 nucleotides is a polymorphism. So polymorphism in the allelic sequence is of variety types. In that, VLTR is one type of polymorphism, where the tandem repeat number is of different. Now I think you understand what is polymorphism, right? Is that clear? Can I move?
Yes, sir.
Yeah, right. Now let's take this. First, you have isolated the DNA. Second, you have digested the DNA by restriction endonucleases. Then you have taken that peak. You have to subject this to density gradient centrifugation. It is not given here because we need that only. Unnecessary, that bulk DNA we don't want. We want the narrow peak. But that step technically is not given in the sequence which is given in your package or in your book. But you have to understand here, after digesting the DNA by restriction endonuclease, when you before digestion of the DNA by restriction endonuclease, when you isolate the DNA, you isolate only the narrow peak. After the step 1, you take only the narrow peak. You collect only the narrow peak and subject that narrow peak to the restriction endonuclease. The narrow peak consists of satellite DNA that you have to break it down by restriction endonuclease. Then you will get separation of the DNA fragments. You will get separated DNA fragments. Then you have to separate them. All the fragments, you have to separate them. This is not a manual work. This is a machine work. First, isolate the DNA, remove the protein and everything else. Subject it to density gradient, you will get a narrow peak. Take out the narrow peak because narrow peak has repeated sequences, and cut the narrow peak DNA into by the help of restriction endonucleases. Okay, then go for separation of those fragments. Many fragments you have to separate it. There is a simple technique which we use, which is called as electrophoresis. So how does this electrophoresis work? Here we will discuss in the extra information in the KVPV classes, the necessary information related to this. But let me give you the basic information which is related to the NEET exam right now on the board. Okay, here there are some wells which are provided here on the electrophoresis board. Now this electrophoresis board, where we drop the culture or drop the DNA fragments in these wells. Okay. And that means we deposit the DNA fragments in these wells. Then one end of the electrophoresis plate is negatively charged, and one end of the electrophoresis plate is positively charged. And the whole base, this whole base So kindly mute your mic. This whole base, if you look at this is whole base. This whole base means this is all, this plate as a base. The base is made up of agarose gel, so that the DNA fragments can smoothly run over the agarose gel. So it will take not more than 5 to 10 minutes. Show some patience here. Okay, don't simply move out of the class. We are at the edge of the topic. So this is what agarose gel, right? So on the agarose gel, DNA fragments can run. How they will run? What is the charge of the DNA? Can anybody tell me what is the charge of the DNA? Positive or negative?
Negative, sir.
Negatively charged DNA, when you subject it to electrophoresis and it is placed at the negative electrode side or negative end side. So this negatively charged DNA run towards the positively charged pole. So when it runs, the smaller fragments of the DNA will run faster than the larger fragments. So the larger fragments will be well behind the smaller fragments. If you look at, we have separated the fragments based on their length on the electrophoretic gel. This gel is agarose. Now, this technique we also call it as RFLP. What is R? R is restriction. Why you are saying restriction? Because I cut the DNA by restriction endonuclease. F is fragment. I cut the DNA by using the enzyme and I made the DNA into fragments. Because of that, the DNA fragments are of different length. That's why I have written length. Different length. I use a word called as polymorphism. Here also I have used a term polymorphism. Now this DNA is showing polymorphism in which case? Length case. In which attribute? Length attribute. So that's why this technique we call it as RFLP. Because of this RFLP is nothing but separation of the DNA fragments by electrophoresis. The separation can be done after cutting the DNA with restriction endonucleases. Due to this, you will get smaller, larger, and mixed fragments of various lengths, and those are placed in a well and placed in agarose gel of electrophoretic channel. And as DNA is negatively charged, these wells are arranged at the negatively charged electrode, and when the electrophoresis is run, the negatively charged DNA will run towards the positively charged electrode. During the run, the smaller fragments will run very faster and the larger fragments will be behind them. Here, I have separated the DNA by its length. That's why restriction fragment length polymorphism. So no more this thing here. I'm not going to discuss anything here. So this is simple technique. Actually, this technique we are not using now. This was a very old and very fast technique that was initially used. Now everything is automated. Put the DNA in the automated DNA machines, everything you will get. Oh, the total thing you will get. You will get the DNA fingerprints also. You will get the number of repeats also. No need to do all this process. So they have a very in 24 hours you will get to know. The paternity issues can be resolved in just 24 to 48 hours. So the technology has developed in that way. But this is the very first and old method which is given in your books, right? Now go to step 4. In step 4, now what we do, we transfer these DNA fragments which we have got, this DNA fragments which we have got onto the synthetic membrane. Transfer the DNA fragments onto the synthetic membrane. Which is made up of nylon. This synthetic membrane may be made up of nylon or which is made up of nitrocellulose material. How do we transfer? Can you take out the DNA with the help of manually? You know, manual cannot be done. So what you have to do, you have to take a nitrocellulose paper or the nylon sheet and press the nylon sheet on the agarose gel. When you press the nylon sheet on the agarose gel, the DNA fragments will be attached to the nylon sheet, or if you use nitrocellulose sheet, they will be attached to the nitrocellulose sheet as it is in the same order in which they are separated on the electrophoresis plate. Now the step 5 is going to be very important here. One second, let me write this down. So you have separated on the nylon sheet. You got a nylon sheet. So let me give you the nylon sheet here. I will take here. I have taken 3 wells there, but I am taking only 1 well. What is meant by 3 well, 1 well? I'll tell you. 3 wells means this is well 1. This is the sample of a suspected person. This is the sample of a suspected person. This is the sample of the criminal. Okay, DNA sample of the criminal. So A, B are suspect, C is the sample of the criminal. Okay, I think this criminal sample, still we can not able to identify right now, but now these are taken out. So this particular A sample is taken on a nitrocellulose sheet. B sample also taken on a nitrocellulose sheet. C can also be taken as a nitrocellulose sheet. I'm just giving you the idea how they do it practically. So I will take one particular A sample onto the nitrocellulose sheet. Just a minute here. I'll take A. So this is nitrocellulose sheet. So on this you have DNA. These are double stranded or single stranded? Double stranded. So you have to break them and make them into single stranded. Actually, you have to break down the bonds, hydrogen bonds, and you have to make them into single stranded before going for this step 5. Okay. Right. That that is an internal step. I'm just giving you an idea. Now you have to hybridize them with labeled VNTR probes. What is hybridize? With labeled VNTR probes. A VNTR probe which is already labeled means it is radioactively labeled. Label means you made it radioactive so that you can identify that particular probe. What is labeled VNTR? Where do you get the VNTR probe? So we have already did Human Genome Project, students. We know the whole genome and we know 60 loci. Already I gave you 60 loci. We have VNTRs. So what I will do, I will collect all the 60 loci VNTR. Sequences are same, no, the number is different. So what I'll multiply, I'll amplify those 60 loci repeated sequences and I'll keep it with me. And I will label it so that whenever any sample will come, I will put this labeled VNTR in that sample. Then this labeled VNTR, if it is here, for example, I made it single stranded now this labeled VNTR will go and bind onto the single stranded where they are complementary to each other. It will go and bind onto it. 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 So wherever they bind, that is the repetitive sequences. Here they go and bind onto it. They go and bind onto it. This is called as hybridization. This is called hybridization. Hybridize them with labeled VNTR probe. Or you can also say hybridization using labeled VNTR. You may ask, sir, how will you know that these are the VNTR probes? I already told you, we have already sequenced the human genome. So we isolated, we amplified, and we keep record of those VNTR sequences. That's the idea, simple idea. So you have all, and I made it, labeled means here, the meaning means radioactive nature is given to them. So wherever they go, you can easily identify. Right, you have to understand this. So here, it's not like, very small piece of DNA you will get in your hand. Know that, very small piece of DNA you will get in your hand. Very small. The sample has very small piece of DNA. You have to isolate the repetitive sequences. Right? When you isolate the repetitive sequences, now you have to, what you have to do, you have to go for what we call it as PCR technique, which you will study again in the zoology, which is called as polymerase chain reaction, where it will duplicate the DNA fragments. It will increase, amplify the DNA fragments. So the sample get doubled. So you have a very good amount of sample with you. Right. Then first step, can we repeat it to avoid confusion? First step, anybody from the class? What do you have to do? Hello, please.
Isolation.
What is the second step?
DNA by restriction endonuclease.
Third step.
Separation of DNA fragments by electrophoresis.
Electrophoresis. Fourth step.
DNA fragments onto synthetic.
So this transfer of the DNA fragments onto the synthetic membrane is called as Southern blotting. And electrophoresis is called as RFLP. Electrophoresis is nothing but restriction fragment length polymorphism. And this, blotting the DNA onto the nitrocellulose paper is Southern blotting. Northern blotting, Western blotting, Eastern blotting will also be there. That is, if you isolate DNA, that is Southern blotting. If you isolate protein, that is Northern or Eastern blotting, like that. But right now, I will come to that point later. First, let us understand the basic steps. It is Southern because we are blotting the DNA onto the nitrocellulose paper. So that's why it is Southern blotting. After Southern blotting, we are hybridizing them with labeled VNTR probes. Then after hybridization, what I'll do, I'll take out this X-ray paper. I'll take out this X-ray paper and I'll put the X-ray paper onto it. This is the X-ray paper. X-ray sheet. When X-ray sheet is kept on the labeled VNTR, hybridized labeled VNTR probes, they are labeled with radioactive. When they are radioactive, they make some bands here. Here it is radioactive. So on the X-ray sheet, you will have a small band formed in there. Take out the X-ray sheet after some time. That X-ray sheet is nothing but the DNA fingerprint. You keep the X-ray sheet on that labeled VNTR, hybridized labeled VNTR probes, okay, for some time. And that technique is called as autoradiography. First, you have to detect where the DNA fragments are hybridized. How will you detect? You don't know. So you have to put the X-ray sheet on that particular nylon or nitrocellulose sheet. Then after some time, you take out the X-ray sheet, you will get the bands. The band is nothing but the bands which are seen on the X-ray sheet is the DNA fingerprint. So the step 6, I think I have given you a clear-cut idea. The detection of hybridized DNA fragments by autoradiography. So each and every step here, that's what I'm saying. These are brief points of the DNA fingerprinting. First step isolation, second step restriction enzyme, third step electrophoresis, fourth step Southern blotting, simply you can say. Fifth step hybridization with labeled VNTR probe. Seventh step autoradiography. I think sixth step autoradiography. Right? Done. Then after autoradiography, you will get the X-ray. That X-ray is called as autoradiogram. So the autoradiogram is called as DNA fingerprint. any doubts regarding this, please shoot now. We have 3-4 minutes of time, no problem. Right, any doubt regarding this? What is the first step? Isolation. Second step? Restriction. Then third step? Electrophoresis. Fourth step? Southern blotting. Fifth step? Hybridization. Sixth step?
Autoradiography.
And over. You have done the DNA fingerprinting. Then after autoradiography, take out the X-ray sheet. Now you will get an X-ray sheet like this, and you will get the bands. You will get the X-ray sheet. If this is the X-ray sheet, you will get the bands. One second, I'll just make these bands a little thinner. This is the DNA fingerprint of an individual. This is, we call it as DNA. The bands wherever they are formed, it means there is the hybridized DNA fragments. So wherever the hybridized DNA fragments are there, you will get the bands. So I'm not going in detail because this is enough for this particular level. So we'll discuss in the extra information wherever necessary inputs are required. So with this we are done. Then one more diagrammatic representation is there. I just wanted to share the diagrammatic representation. Then you will get some idea on that. Okay. So let me go with the diagrammatic. So then with the help of that, so in the next class we will be starting strategies of enhancement. Before strategies, I just wanted to go for some extra information. Then after that I will start strategies. Look at, this is one example that I can give you. Here, so this is what the so this is a DNA fingerprint of A. This is a DNA fingerprint of B. This is A and this is B. Very clearly given here. Look at paternal chromosome, maternal chromosome. Paternal chromosome has only 3 repeats here. Maternal chromosome has 4 repeats. Look at here. Chromosome number 7. This is of A. But look at B. This side is B. This side is A. The paternal chromosome has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 repeats here. Okay. And the maternal 1, 2, 3, 4, 5, 6, 7, 8, 9 repeats here on chromosome number 7. So pretty easy to identify. If you look at this C, I think this is C is the DNA from crime scene. So on this paternal chromosome 11 and 9, if you look at chromosome 2 also, the same number are there here. In chromosome 16 also same number are there. So DNA from individual B and DNA of C both are matching. So the culprit is B. Here number of short tandem repeats are given here very clearly, right? Danish, I will call you within 5 minutes. Sir. Right? Is that clear? So this one will give you some idea. Once you work out with the diagrammatic representation, see this B is matching with C. Is the C. Okay, this is the way they identify, they use it in forensic applications. This is actually the representation of the DNA fingerprint. In this way, the DNA fingerprint is right. We'll do some questions on this in the next class, and we'll do some more info. I'll share some more information related to this. Then I'm done with the molecular analysis. Thank you very much. Have a nice day. Any doubts, you can stay back.
Sir.
Akshay, tell me.
Sir, I cannot understand the step of hybridizing the VNTR with the DNA sample.
Okay, can you let me know? Do you understand what is hybridization?
Sir, no, sir. That only I wanted to know, sir.
Hybridization is nothing but formation. For example, there is a sequence called as A, C, TTAC, right? Now I have a what we call it as a probe, a VNTR probe. This is a VNTR probe, right? This is a VNTR sequence which is present on the DNA fragment which I have placed here, right? And there is a probe which is coming and binding onto it. It should be what? This is T, this is G, this is A, this is A, this is T, and this is G. So these probes I have inserted here. Now this probe will go and bind onto this. This probe is radioactively labeled. So this probe is binding onto this particular DNA which I have introduced here. This black color is the DNA which I have introduced. Okay, and they are present on the nitrocellulose paper based on the length. Then when I introduce the probe will go to the complementary sequences and bind onto the complementary sequences. So this is the radioactive probe. I have written R. This radioactive probe sequence is this. This will go and bind onto this particular sequence. So this is called a hybridization. Formation of hydrogen bonds is nothing but hybridization. Now, what is your doubt?
No, sir, clear, sir. I could not understand how would that hybridizer now clear, sir.
Not clear, no? Right. Because I already told you that the DNA which is placed on the electrophoresis flannel will become single stranded. The hydrogen bonds are break down and they become separated and they become single stranded, so that they are accessed by VNTR probes later in the step. Right. Please ask. Right, I'm done. Any doubts you can ask. Lakshanya.
Sir,
Akshaya, tell me.
sir, one more doubt, sir. Sir, if monozygotic twins in that case, or in all 60 loci, they had same VNTRs, no, sir. Then if one of the person does some crime, then how will be able to differentiate between them, sir?
How will be able to differentiate? You see, during meiosis, it's only can be able to differentiate by the help of the sperm they produce. Maybe the sperm, whatever the sperm cell that they produce, will not have the same, sequence. That means what we call it as they are not be identical, right? Because they are formed by meiosis. Right? There is a change in the gamete. So only the sperm cell that they produce will be different. Right? Yes or no?
Yes, thank you, sir.
Thank you, Akshay.
Sir,
Lakshmi, you are asking about Shivani, you are asking Ajitesh, you are asking. Okay. Ask.
sir, no, sir, if monozygotic twins have exactly the same 60 loci, sir, then how they have some externally different features, sir?
They won't have external different features now. They have same features only.
So, no, sir, sometimes we are able to distinguish between them, no, sir.
I don't know whether we can able to distinguish by continuously talking to them the way they talk, behave, and that only, not morphological differences.
Yes, sir.
Because see, the only thing, Ajitesh, the fundamental thing, that is only one zygote now, Ajitesh. One zygote means this is the male gamete and this is the female gamete, and this is one zygote. So this zygote is dividing, means these cells which are present in this zygote part, these cells which are present in this zygote part are both are same only. So how do you identify the difference between these two? Because these are formed from the same zygote. Actually, monozygotic means the zygote divides and two parts will behave individually, completely as a whole, right? It is not dizygote, it is a monozygotic twin. Two individuals are formed from the same zygote. So there is no difference. If dizygote means definitely there will be a difference. Monozygotes, no? One zygote.
Yes, sir.
Right.
Yes, sir. Thank you, sir.
Thank you. Lakshanya? Do you have any doubt, Lakshanya? Right. Yasir, tell me. Tell me. Yes, Ram? Yes, autosomes. One second. Allosomes or autosomes, it's not that. If the mutation occurs in germ cells, the mutation may occur in autosomes or allosomes. It may be whatever. Say why? Because in a gamete, don't you find autosomes in a gamete? You find autosomes also, Nara. Yes, sir, in a gamete. Then this is about mutations occurring in germ cells. Only 6 chromosomes they don't have, Nora. They'll have autosomes also, no, one set. You are missing the point. They will have autosomes, Nora. You have 46, they'll have 23. 23 means only 6 chromosomes. No, no. One 6 chromosome. They'll have either X or Y. And the remaining. Yeah.